My real pleasure to introduce the first speaker, Professor Giovanni Romano from the University of Florence. Giovanni, who will be describing the physics behind Light4Lungs. Giovanni, thank you very much. Please.
Thank you very much. I have to say it was a real pleasure to participate to this project, and I hope I will convince you that the physical aspects of the project were properly understood and studied. So I represent here the University of Florence, of course. The work I will present to you has been done together with one of the third parties of the University of Florence, which is the University of Siena. And I also represent the collaboration with the Sorbonne Université from Paris. Okay, so let us start by reminding all of us, that was already introduced, which are the species that we considered to be most representative of lung infections. And those two species are, as you can read, Pseudomonas aeruginosa and Staphylococcus aureus. And of course, you know also that once the species is fixed, you may have different strains, and these are the names of the strains, respectively of one and the other, that were chosen in the project to be studied from the in vitro experiments up to the in vivo experiments, which you will ex vivo, which you will see later on. I would like also to briefly remind you, as we will be dealing with results obtaining on a biofilm, that of course you can have bacteria in the so-called planktonic state, so bear with me, especially the microbiology and biology components. As a physicist, this is what I have understood from collaboration from the project. So planktonic means that the bacteria are in a liquid medium and they are, so to say, free to be dispersed in that medium. But of course, if you leave them the possibility to attach, especially to attach to a surface, so according to the conditions, what they will do is that they will attach to the surface and develop a community which is called the biofilm, which is formed by bacteria, of course, but also of substances produced by bacteria like sugars, DNA components, and proteins, and so on. So that environment allows them to live well and to be more protected than the bacterium itself, the bare one, let's say. So if we have to resume what we wanted to answer as our contribution, the question we wanted to answer was that one. So how the different light colors are effective in photoinactivating bacteria? This question has an answer which can be easy or difficult according to the system you are studying. And what we want to analyze is thinking of the final conditions. So when I want to illuminate bacteria, the biofilm in vivo, those are the conditions which are the model the conditions I have to model to answer that question. Finally, answering that question means to find a curve like this one, which represents the efficacy in obtaining a photo inactivation effect as a function of the wavelength of the light you shine. This corresponds formally to know the spectrum of action, so the action spectrum for photoinactivation in the conditions that we have chosen, which correspond to the in vivo conditions, so in humans. We did not perform experiments on humans, but what we did is to measure optical properties and in vitro results of photoinactivation, developed a model, merged all data, and obtained that curve in terms of a prediction. That prediction was important to inform the other partners, especially in charge of particle synthesis, development, and characterization, and to tell them which colors had to be contained in the emitted light by the particles. Okay, so the objectives are actually two. The first one was to demonstrate that light can kill bacteria in the biofilm state, and to predict which light colors have the highest antibacterial efficacy when applied in vivo in the lungs. So this information was passed on to the project partners in charge, as I said, of particle synthesis. So the very first model you could have in mind is that you shine light onto your petri dish, like your system, your container where the bacteria are, for instance, on a surface. So this is what I wanted to depict by this cartoon, where you have the light shining particle of our aerosol directly illuminating our bacterium. Actually, this is too simple a model, because in reality, as I said, at least you have to consider that light is shined onto the biofilm. So, but bacteria are inside it, and you have to understand what happens when light interacts with the biofilm, because that changes possibly, and we demonstrated that it does, changes the intensity and also partly the color of light when it arrives to the bacterium. More complex, third model, which is more realistic, is that you have to add the lung tissue. So everything happens in vivo. So not only you have to illuminate to arrive to deliver our photon cargo in the bacterium cell, but you have, which is in the biofilm, but first light will be shined onto the lung tissue. So we have also to understand the influence of the geometry and of the lung tissue optics on light, which makes things even more difficult, but that's life. So if you want to go in vivo, you have to face these kind of problems. Then the methodology that we used. You remember the first question was, can light kill bacteria? As was anticipated, we rely on the presence inside bacteria of photosensitizers, which are porphyrins. We then designed an experiment to shine light onto biofilms in laboratory conditions. What we did is to buy LED sources, because we needed a light source before, of course, designing the aerosol, and we chose violet, blue, and green, and a red light source to cover the entire spectrum of light. Then the geometry is the one you see on the left-hand side. We illuminated from the top, and what you see is what is called a multi-well plate. So a container, let's say, where you have your biofilm in a proper medium. That is in the center a real image of the illumination conditions with the blue light, and the final image represents on top the conditions where we have grown the biofilm. So you see those on top, those little plastic fingers. So they are inserted into the wells, and in the wells you have bacteria. They grow a biofilm around the finger. When the biofilm is ready, you reverse it and you illuminate from the top. Okay, this is the geometry. What we did is to vary the light color, and we also varied the species and the strain. Then the results relative to this setup. What you're seeing here was anticipated before, graphs representing the photoinactivation efficacy versus the so-called light dose, which is in this case represented by the energy that arrives onto the first biofilm surface per unit surface. So you could transform it in terms of how many photons per square centimeter arrived during the irradiation time. Okay, so as you can see, in general, the higher the better in this case, because the higher is the curve, the higher the efficacy in photoinactivating. This is the difference between how many bacteria are counted alive after the treatment and before the treatment. So there's just some mathematics. But the basic thing is that the higher the better. So you can see that according to the strain, the effect is different. And in general, if you fix the strain, the effect is different for different doses. And it is what you think, probably. So the higher the dose, the higher the number of photons delivered, and the higher the efficacy. So that efficacy is changes according to the strain. And in this case, I show you the violet light irradiation. In general, as I said, we consider the 4 colors. So this is the a panoramic view of the results of the 4 colors. And what you can notice is that the violet is the irradiation corresponding to the maximum efficacy, because even if I don't go into detail, you can see that blue is less effective, so curves are more flat, and even worse, let's say, with the green and the red. I remind you that this is light directly onto the biofilm in laboratory conditions. So this is a first result, which corresponds to the very first model of biofilm without taking into account the tissue. The collaboration with Paris and Siena was from 2 different points of view. So what you saw before were in vitro experiments. We wanted actually to couple them with a microscopic approach. So we can resume by saying we had a macroscopic, so a big approach, meaning we had macroscopic samples which were eventually illuminated and counted, but also microscopic with microscopy, optical microscopy, and millifluidic techniques. In this case, what we did is to study a growing biofilm. Biofilm, bacteria are alive, they multiply, they grow in number. So what you want also to understand is the timing and their density and the response to light when you actually can see them in the microscope. So this setup, simplified setup, is to acknowledge the presence of light interrogating your sample, and I will show you in a moment the results. As I said, this is a microscopic, but the macroscopic approach started with the results you saw before and continued, because as you will remember, after the light onto the biofilm model, we have light, tissue, and biofilm. So how to take into account the tissue? Then what we did is to consider, as a physicist, you know, I like simple geometries like spheres and cylinders, and actually you can mimic the upper airways like the trachea, bronchioli, with a cylindric geometry where the emitter is the single aerosol particle. What we understood is that if you consider the presence of an infection, our model considered mucus and biofilm. The B and the M are biofilm and mucus mixed together, and light is shined onto this layer inside the lumen in this or this other geometry. Then what we did, and I will skip some details, is that we wanted to simplify this very complex problem. Then the simplification was in terms of what if we could reduce this problem to a monodimensional one, which is an abrupt approximation, but it took a lot of time, even if it is a one-dimensional problem. So we succeeded in saying, from the point of view of the interaction between light and the tissue, how, if I want to have a one-dimensional problem where we have only some tissue and biofilm, and then some tissue and then all the bacteria at the bottom. If we could reduce everything to this model, then here I can simulate the optical properties of this and infer how light is changed by the passage through the tissue, and then understand what I have to do in reverse to have to maximize the photokilling here. So I realize I'm skipping some passages, but the message is that we eventually simulated the interaction of light with a very simpler model, and we were able to measure the action spectrum. Let us have a look at the results from a microscopic approach. So there you can see an example of channels with biofilm inside. You see the glove of the experimentalist, and in the center image you see how it looks at the microscope, a growing biofilm. You see from top to bottom the density increases. And then here is an example of a measurement where you monitor in time the increase in the population, so in the density of the biofilm. This is in terms of optical density, which you can correlate with the density of cells. Okay, so we were able to follow what happens at this level. And here is a short film. Not sure if I can, maybe from here. Thank you. So you see a time lapse of what happens when you start a biofilm. So you see the density grows and grows. Of course, here we see especially the cell component, but this is what happens in time. So you have to think of the process, a dynamic process. You are irradiating, but they are alive, they are dynamic, and they change their properties. So finally, merging all results together, this is the result we obtain in terms of action spectrum. The y-axis is in arbitrary units, which means 1 corresponds to 100% of efficacy, arbitrarily defined, because what you have interest in measuring here is which is the relative efficacy of two different wavelengths. So if you consider 400, 410, that is the maximum respect to every other wavelength, okay? In the x-axis, of course, you have the wavelength. You can notice the presence of different curves which are very similar to one another. Why that? With careful analysis of the literature, you of course understand that each patient is different from another one, so it is not that you don't have one condition only. You may have thicker or thinner biofilms. You can have the presence of 1, 2, or more bacterial species. In vivo, you can have more or less oxygen and so on. So the different conditions were modeled with parameters in the model, and those parameters led to slightly different results. We didn't know a priori the result, but we were just also, let's say, a little bit lucky in terms that all the curves are very similar to one another. So this result is quite robust and indicates that we have better illuminate with the violet blue light in all conditions. Of course, this is a prediction and has to be confirmed by in vivo measurements. So this is the main result with the work, part of the work performed by Florence, Paris, and the University of Siena, and this result was passed on to the other partners for particle synthesis. So was forgetting that I have the take-home messages for you. So visible light can photoinactivate bacterial biofilms, and the efficacy depends on the strain, on the light color, and on the dose. Second, the best predicted conditions to maximize the action of light in the lungs correspond to a violet-blue emission for the aerosol particles. So they have to contain that part of the spectrum to maximize the efficacy. Thank you.
Hello, good morning. Thank you for the comprehensive talk. I am new in this topic and really enjoyed your speech. I have a couple of questions, a very related one with the other, and it's related to the conditions of the tissue. Do you plan, or maybe you have already done it, understood, do you plan to model also the temperature of the tissue that may vary? This is first question. And second, since now we have a lot of patients with fibrosis, no, kystic fibrosis, do you also plan or have you already done the model with the tissue with fibrosis, or do you or it doesn't make any sense? Maybe they are silly questions.
Absolutely. Thank you for your questions. No, I mean, we had a lot of questions ourselves. So regarding the first one, we considered to model correctly the geometry of and the physics behind the interaction, the conditions, physiological conditions, like the ones which are associated to the results in literature. So literature gives us some results regarding the thickness and the oxygen concentration and so on, and all those results were related to physiological conditions. Therefore, we did not inquire a variation in temperature. That could be done in principle because there could be some changes in the optical properties. I think that probably that would be a minor correction, but still it is worthwhile inquiring that. So that was not explicitly done. For the second question, so we were inspired especially by cystic fibrosis conditions. We have published those data. We envisage, we think we could also have a better model, but we believe that corrections are really very minor, because even when you talk with a clinician and you ask where is the infection exactly, is it mainly here, there? So the answer is, I would like to know exactly, but actually I have some ideas. But as was also anticipated by the colleague in Liverpool with a beautiful talk, clinical talk, they don't know precisely. So it is also a problem of information available.
Thank you. Thank you, Dr. Giovanni, for your nice talk. I have two questions. The first one is regarding the LED sources exposure. The exposure time of the LED source was, which was the exposure time? Because if I'm not wrong, you activate the nanoparticles for 1 minute or so, and then they are active just for some seconds. So I wanted to know the exposure time. And the second question is about the last question is about cystic fibrosis. We know that the mucus accumulation is thicker. So I wanted to know if you have done some calculations about or regarding the deeper that you can go or you can be active with your therapy. Thank you.
Certainly. So the first question, those experiments I showed you before were done as preliminary experiments to understand how the aerosol had to be synthesized. Therefore, we used LED illuminators, so the same kind of source as the LED for house illumination, peaked in some colors. The illumination time was in between 15 and 40 minutes, more or less, to achieve the maximum dose that we wanted to achieve. The second question, so the cystic fibrosis problem. So yes, we considered data from the literature. When we modeled the biofilm, we had two extremes, a minimum and the maximum thickness, which was taken from the literature, and the maximum, as far as I remember, that was of around 0.5 millimeter, maybe more, depending on where exactly. The penetration depth of light into the biofilm mucus was varied according to the conditions. It could be of around in between 100 and 300 microns. So that was compatible to reaching all bacteria, though of course with different intensities.